Hello viewers, welcome to this chemistry practical analysis video. In this video lesson, I have analyzed the qualitative analysis aspect of the 2024 NECO Chemistry Practical Examination step by step to show you how to identify unknown ions present in a sample so that you can pass your chemistry practical examination with ease. So to get the best out of this video lesson, do well to get your writing material and do not skip any aspect of this video. Before we look at the various samples for the qualitative analysis aspect of the 2024 Chemistry Practical Examination, here are certain tips that should be noted about the practical examination. The practical examination is divided into two parts, which are the quantitative analysis, that is the acid-base titration, and then the qualitative analysis, which is salt analysis. And then you'll be asked a third question, which is a random question from any aspect of chemistry. It means that you do a first analysis, usually acid-base titration, that's your first question. Then your second question will be a qualitative analysis question, that is a salt analysis. Then you will be given a third question, which is a random question. Probably you could be asked, what is deliquescent, or list 3 drying agents. So these are the two samples for the qualitative analysis aspect of the practical examination. We have sample C as zinc chloride and we have sample D as powdered milk. And in each of these samples, we are testing for the presence of certain ions or certain radicals. For example, in sample C, we are testing for the presence of zinc ion. Then in sample D, we are testing for the presence of protein, testing for the presence of amino group, and also testing for the presence of carboxylic group. Let's get started. So begin with sample C, which is zinc chloride. The first test you'll be told to carry out is to add sample C to a given volume of distilled water. When you do that, sample C will dissolve. So under observation, simply write sample C dissolve to give a clear solution. That's what you can observe. Then from what you can observe, what can you draw out or what can you infer? So under inference, right? Since it dissolves, it means it is soluble. So simply write sample C is soluble. Next, they could tell you add a blue litmus paper into the solution or dip a blue litmus paper into the solution. When you do that, you observe that the blue litmus paper will turn red. Why? The reason is simple. Zinc chloride is made from a strong acid and a weak base. So when we dissolve zinc chloride in water, it will form a weak acidic solution. So when we dip the blue litmus into that solution, it will turn red. So therefore can infer that sample C is acidic. Then they could tell you to divide that solution into two portions, like divide them into two different test tubes. So they could tell you to work on the first sample. So in the first sample, they could tell you add a given volume of aqueous sodium hydroxide in drops. So in drops, like you can just put 2 or 3 drops. When you do that, you observe a white gelatinous precipitate. It is gelatinous and not chalky. So don't ever make that mistake of writing a white chalky precipitate. It's a gelatinous precipitate, just like what is displayed on your screen. So from that observation, what can we infer? We can infer that either zinc ion or aluminum ion is suspected. Next, they could tell you to that solution above. That's when we have added sodium hydroxide in drops. They can tell you to add excess aqueous sodium hydroxide. When you do that, the precipitate will dissolve to give a clear solution. And from that, we can also infer that zinc or aluminum ion is still in that sample. So we are suspecting either there's zinc ion or aluminum ion. Then they could tell us to the second portion, add a given volume of aqueous ammonia in drops. When we do that again, we will get a white gelatinous precipitate. And from that observation, we can infer that either zinc ion or aluminum ion is suspected, because those are the two ions that can give us a white gelatinous precipitate. Now, the last test is what we call a confirmatory test. So when we add excess aqueous ammonia to the solution above, that is the one we added aqueous ammonia in drops, the precipitate will dissolve to give a clear solution. Only zinc ion will do this. So because it dissolves, we can therefore infer that zinc ion is present or zinc ion is confirmed. So that's all for sample C. They won't ask you to test for chloride ion because the reagent needed to test for chloride ion, which are silver triazonitrate 5, that's AgNO3, and then nitric acid, that's HNO3, will not be provided on the day of the examination. So let's move on to sample D. Moving on, sample D is a powdered milk. So in the exam, you'll be told to divide this sample into 2 or 3 portions. So when you're told to divide into 3 portions, it means divide that sample into 3 test tubes. So to the first portion, you could be told to dip a litmus paper into it. Now what we observe, naturally the powdered milk has what is called lactic acid, meaning we expect that it should be acidic. But during preparation in the industry, it is made alkaline to prevent a disease condition known as acidosis. So because of this nature, when we dip a red litmus paper, it turns pale blue, not completely blue. So because it turns pale blue, it means sample D is slightly alkaline. Next, they could tell that to that solution, add a certain volume of aqueous sodium hydroxide and a certain volume of copper 2 tetrasulfate 6. When we do that, the color of that solution, which was blue, will turn pink or violet. Now, when we get this coloration, it simply tells us that protein is present. And this test is known as the biuret test, and the biuret test is used to test for the presence of protein. So the biuret reagents are NaOH, that's sodium hydroxide, and CuSO4, that's copper 2 tetrasulfate 6. To the second portion of sample D, you could be told to add soda lime solution and then heat it. When you do that, you'll find out that the solution turns cloudy. Why is it cloudy? Because there are some insoluble components. So under inferior, simply write presence of insoluble component. Then to that solution, you could be told to use a filter paper to filter. When you filter, you find out that there's a pale white residue that is gathered on the filter paper, and then the filtrate that passes through is clear or colorless. That also signifies that, or from that, we can infer that there's presence of insoluble component. So simply write under the inference, presence of insoluble component. Next, they could ask you to that filtrate that you've obtained, add a litmus paper. When you add a litmus paper, a red litmus paper to be specific, the red litmus paper turns blue. Since it turns blue, it tells us that the filtrate is alkaline. Also, you observe that, or when you perceive, you find out that the filtrate has a fishy smell. And a fishy smell is a characteristic of the amine functional group. So we can simply write, because of that fishy smell, we can infer that the amine functional group is present. Next, moving on, they could ask you to the residue, add a particular or a certain volume of dilute HCl. When you do that, you observe that there is bubbling of gas, and bubbling is what we call effervescence. So there's a bubbling of a colorless and odorless gas, and that gas turns lime water milky. And one of the gas that can do that is carbon dioxide. Also, the gas turns a damp blue litmus paper red. So because it turns a blue litmus paper red, we can infer that the gas is acidic. Secondly, the gas turns lime water milky. We can also infer that carbon dioxide is evolved. Also, in this sample, the sample D contains both amino acid and also a carboxylic acid group. The reason is simple. Let's come back to the second portion. When we add soda lime, the function of soda lime solution is to break down amino acids to give us three things. It gives us amine. Amine is the alkaline part which passes as the filtrate. It also gives us CaCO3, that's calcium triazocarbonate 4, which gives us the CO2, and then water. The CO2 is the acidic portion. Then lastly, to the third portion, the octator to the third portion add a given volume of HCl. When we do that, we find out that a white precipitate is formed, and that confirms the presence of a protein. Simply, milk is proteinous in nature, so Therefore, everything we are trying to test here is to test for the presence of protein as well as the main functional group. Thanks for watching. I hope you learned something from this video. Don't forget to like and share these video lessons. Also, do it to subscribe to this channel if you haven't done that, so you always get notified as soon as new videos drop on this channel.